Vyriad has done to develop a clinical test that's specifically going to detect neutralizing antibodies against the COVID-19 virus. And for those of you that aren't familiar with Ammonis or Vyriad, we are biotechnology companies that are interested in developing new oncolytic viruses for treatment of cancer. And one of the main platforms we use for this is VSV. And so when the COVID pandemic came, we wanted to see if we were able to use that VSV platform in order to make some tools that would help combat what was going on. So I'm going to start with kind of the required introductory slide, but I don't think it's very necessary because most people are pretty aware of what's going on with COVID. Essentially, it's caused by the SARS coronavirus 2 virus, and there have been 2 previous human outbreaks of coronavirus that we know of, including SARS coronavirus and then the Middle East respiratory syndrome. There's a lot of things, obviously, that we don't know about COVID and a lot of important questions that we're still trying to answer. And these are just a few of those questions as they relate specifically to immunity and protection. And so, for example, what are the rates of immunity in populations? What constitutes a protective response against COVID? And if you get a protective response, how long is that response going to last? Is reinfection possible? Do you need to worry about that? When we look at potential vaccines or treatments, how well are these vaccines or are these treatments at providing a protective immune response that's ideally also long lasting? And so with these questions, a lot of them depend on having good serological tools in order to examine COVID immunity. And for those, there's essentially a few different types of serological tests that are available. There's the ELISA test, which can be qualitative or quantitative. It's a rapid test that detects the ability of antibodies from a sample to bind to different virus antigens. There's also these lateral flow rapid diagnostic tests that also are going to detect the ability of antibodies from a sample to bind to virus antigens. So the important thing about both of these tests is while they are rapid and relatively easy to use, they are only able to detect antibodies that bind to the virus, and they are not specific for antibodies that neutralize the virus. And this is an important distinction because while a lot of antibodies can bind, only neutralizing antibodies are capable of blocking virus infection. And so in order to look at neutralizing antibodies, we use a plaque reduction neutralization test. And this test will detect the ability of antibodies from a sample to block virus infection of cultured cells. So while the PRNT assay is the gold standard for detecting virus neutralization, there are a few limitations. Mainly, it has to be performed for COVID in a BSL-3 lab, so there's a safety concern. There's also a limited number of labs that are able to do those kind of tests. And also, traditionally, these tests are done where you have to count plaques manually, and so they tend to be relatively low throughput. And so the assay works well, but it doesn't work very well when you're trying to do large-scale analyses of neutralizing antibody levels in populations. And so we wanted to see if we could use the work that we're doing in our lab to try and combat this issue by making a safer, scalable alternative to the traditional PRNT assay. And this is an overview of the assay that we developed, and it uses 2 key components. It uses an engineered artificial mimic virus. This is based off of VSV, and I'll go into a little more detail about that in a moment. But basically, this mimic virus can infect cells like the COVID-19 virus. And so we can use it to see what samples are able to block virus infection. And then we also use an engineered reporter cell line that is able to detect virus infection through a luciferase signal. So if there are neutralizing antibodies present against COVID, they would bind to the mimic virus, there's no infection and no detectable signal. Whereas if there aren't neutralizing antibodies, there is infection in the cell layer and we get detection of signal. So for those of you who aren't familiar with VSV, it primarily infects horses and cattle and swine. Natural human infections are rare and they are typically asymptomatic or have very mild symptoms. It's a negative sense RNA virus that is composed of the viral RNA genome along with the RNA polymerase and a phosphoprotein that then is coated in a nucleoprotein and a matrix protein. And this is all in a lipid envelope that's derived from the host cells, which contains the membrane glycoproteins. And those membrane glycoproteins are what mediate binding to cells and infection of the virus into the cells. So what's great is that there are several established reverse genetic systems for engineering recombinant VSVs. So it's very common to make these payload viruses where different genes are inserted into different places in the VSV genome so that the viruses will express them. But the other really nice thing is that the VSV glycoprotein G can be replaced with glycoproteins from other viruses that facilitate cell entry. And so, for example, in VSV EBOV, the Ebola vaccine, the VSV G protein was replaced with the Ebola virus glycoprotein, and that was used as the basis for the vaccine. And so we have a lot of expertise in this VSV engineering, and we wanted to use a similar approach to try and make this mimic virus for the assay. And so what we did is we took the VSV-G and we swapped it out with the SARS coronavirus 2 spike protein. That's the protein on SARS coronavirus 2 that allows it to enter cells by binding to the cellular ACE2 receptor. We had a deletion at the C-terminal region of that spike protein because that allowed for better incorporation into the virion particles. We were able to confirm by Western blot that the spike protein was specifically being expressed in our VSV spike virus. And we detected both the full length and cleaved variant of spike, and we did not see any G expression in our spike virus, though we were able to detect the other VSV proteins still. So then, of course, the question is, now that we have this virus, how can we detect it in a high throughput format? And to do that, we exploited the fact that the virus will cause cells to fuse. And so cells that express the ACE2 receptor, for example, viro cells, when they are infected, they will form syncytia, or these clumps of fused cells. And there's a system that's been developed called the dual slit protein system that allows for quantitation of the level of fusion that's occurring in cell culture. The way this assay works is essentially there's a combination of two proteins where half of Ranilla luciferase is connected to half of green fluorescent protein. And then two different cell lines will express complementary halves of this combination. So if a cell line that's expressing DSP part 1 fuses with a cell line that's expressing DSP part 2, it will form fully functional Ranilla luciferase, an EGFP, and that can be monitored using a luciferase assay. So we engineered DSP1 and DSP2 viro cell lines by lentiviral vector transduction, and then used them to measure infection using our VSV spike virus. And we saw that indeed, when we infected the cells, we were able to see increase in luciferase activity. It decreased a little bit later, consistent with all the cells basically dying due to the virus infection. So we went on and we optimized the conditions to get better luciferase signal. And we found that as we increased cell number, we saw an increase in luciferase activity and fusion from virus infection. Interestingly, we found that we actually had to decrease cell number from the flasks that we were seeding the cells off of in order to get good expression. And we think this is all related to ACE2 expression levels on the cells. We also found we could seed cells directly from cryopreserved stocks as long as they're given 2 days to recover. Not surprisingly, as we added more virus, we got more fusion and higher luciferase signal, though it did seem that there was some lot-to-lot variability in terms of the virus's ability to cause fusion, and it wasn't directly related to the concentration or the titer of the virus. But then we wanted to say, okay, we've shown that we can detect fusion. Can we actually block fusion? Now, initially, there wasn't a lot of convalescent samples available that we could use to do our testing. So the first things we tested were some purified proteins that were expected to bind to ACE2 or the spike protein, and we would predict would block fusion. And we saw that, yes, indeed, we were able to block luciferase signal from the virus when we use these different purified proteins in a dose-dependent manner. We then were able to get some convalescent samples, and we found that those also, convalescent plasma was able to block the luciferase signal in a dose-dependent manner. And this would be compared to the negative control plasma from an unexposed individual. And again, it was a dose-dependent inhibition, and the lower luciferase activity indicates that we're getting a block of virus infection. So I'm going to go pretty quickly through this slide, but essentially we've optimized the assay workflow in order to improve conditions to maintain virus stability. We found that we were able to reduce the neutralization time from the standard 1 hour at 37 to 30 minutes at room temperature. We've also done some optimization on how to treat plasma samples to avoid thermocoagulation. We are ongoing with work to look at whether or not samples should be heat inactivated. We do have some preliminary evidence that heat inactivation can reduce the sensitivity of the assay, and we think that may be related to some contribution of complement in those samples. And so that is ongoing, though our currently validated assay does not include heat inactivation. So having optimized these different conditions, we wanted to go ahead and see what the clinical correlation was. Was our assay doing a good job at predicting whether or not people had neutralizing antibodies? And so we got a clinical trial up and running, and we also collaborated with John Mills' group at Mayo Clinic, who provided us some additional samples for testing in the assay. And we got samples from 3 different cohorts. We had a positive cohort, which was people who had tested positive for the virus by PCR or positive for anti-SARS-CoV-2 antibodies by another serological test. We also had the negative cohort of people who had either tested negative or had no known exposure or symptoms of COVID-19. And then we had a test cohort, or what we called our contact positives, were people that had been exposed to somebody who tested positive for COVID-19 but hadn't been tested themselves. And so they were kind of our unknown group. Out of the total of 230 samples that we tested 125 presumed negative samples, and all 125 of those samples we detected as negative. And so we didn't have any false positives in this assay. So it was demonstrating, excellent specificity. Of the 72 samples that we expected to be positive going in, we detected 69 of those as positive. All of the samples that had been detected positive by PCR were also positive in detected positive by ELISA were also positive in our assay, but there were 3 that had been positive by PCR that were not positive in our assay. After further investigation, 2 of those were not positive by other serological tests. And we think a lot of that has to do with if the sample is taken too soon after the PCR test, there's not enough time for the neutralizing response to have really amped up yet and be detected yet. And so essentially we are seeing also very good sensitivity with the assay. Of our test group, we detected 13 of the 33 that were contact positives as positive. So overall, we had very strong clinical correlation between what we would expect and what our assay was reading out. But we didn't want to be able to just give a positive or a negative result. We wanted to be able to give a quantitative result, because it's really important with these neutralizing responses to know the strength of that neutralizing response. And so we developed a calibrator system to go along with our assay that was based on a control antibody that binds to the spike protein and inhibits virus entry. And so we can add increasing amounts of this calibrator antibody into pooled negative samples. And we can see that as we add those steep concentrations, the percent luciferase signal that we're getting decreases. And so this allows us to generate a standard curve or a calibration curve. And we use that to calculate something called virus neutralizing units. And we set the virus neutralizing units as an arbitrary unit that's essentially equivalent to that antibody concentration multiplied by 100. So for example, if we had a signal that was out here, that would be equivalent to 30 VNUs. And if we had a sample that gave an equivalent luciferase signal down here, that would be 300 VNUs. And we found that 30 to 300 was roughly the linear range of the assay for the positive samples that we were getting. And for the samples that we looked at, we did see that we had quite a range in different VNU values. So this is a representative of some of the positive samples that we'd assayed. And you can see that there is quite a range in terms of levels. And so this highlights again the importance of being able to quantitate the amount of neutralizing antibodies, because clearly there is quite a degree of weak responses to strong neutralizing responses. And especially if you're thinking about things like what kind of response do you want from a vaccine, or what type of donor might you want to donate plasma for a convalescent plasma therapy program, you're going to want donors that have very strong neutralizing antibody responses, and you want vaccines that are going to induce a very strong neutralizing antibody response. So because this VNUs and this assay is a little bit different, it uses a different virus than the standard PRNT assay, one of the important things we also needed to do was just make sure that the results we're seeing correlate well with what you would see for the actual SARS coronavirus 2. And so we did send off some of our positive samples to the University of Texas Medical Branch, Scott Weaver's group, and they ran a PRNT assay using a clinical isolate of the SARS coronavirus 2 virus. And what we saw is that there was, very good correlation between the VNU values that we were getting and the PRNT EC50 values that were coming from this assay. And so we do think, the assay is very good at reflecting what's actually happening with the SARS coronavirus 2. The VNU values seem to be very, very accurate, but we've gotten some feedback from some of our customers that they're used to seeing virus neutralizing titers or a different type of unit. And so we are also looking at doing kind of an alternate assay method where we measure samples at different dilutions and we report them in more of the standard virus neutralizing titer. It doesn't really change the assay overall that much. It's just a different readout that can be easier for certain people to understand because it matches more with the traditional readout. So just to summarize, we have developed a scalable quantitative test for detecting virus neutralizing antibodies against COVID-19, and it is clinically validated and the EUA is under review. The assay exploits the capacity of the virus to induce cell fusion in a DSP luciferase system in order to get that high throughput readout. And we can use this assay without needing BSL-3 level. For anybody who wants additional information, we do have a preprint manuscript that's available on bioRxives. And at the beginning, I started by saying, there's a lot of important questions we still have about COVID. We certainly haven't answered those questions at all, but I do think this assay helps bring us one step closer to answering some of those important questions. And of course, I have to finish with acknowledgements. This was a huge collaborative effort between Amanis Life Sciences, Viriad, Regeneron Pharmaceuticals, and Mayo. And I'd like to especially point out Samantha Reiter, who, along with me, did almost all of the laboratory experiments that are shown today. And also Tim Carey, who wrote our validation plan and does all of our statistical analysis, and as well as the donors who participated in our clinical trials. And with that, I think I'm out of time, and I think I am also out of time for questions. So I will just encourage anybody who has questions to visit my breakout session after this. And we will now proceed on to our third speaker of the morning, which is Dr. Leo O from the University of Minnesota. And he's going to be speaking about the proprietary PS gene editing system that his lab developed and how they have been using it to treat a number of different lysosomal diseases. So.
Okay. And the story began with our previous efforts with Sangamo Therapeutics. So we developed a zinc finger ZFN-mediated in vivo gene editing and tested in MPS 1 and MPS 2 mice. And these two studies result in 2 phase 1 clinical trials. And that's a first in human gene editing clinical trial. And here's the first MPS 2 patient who received the treatment. However, the progress reports found there's a low level of transgene expression, and you may wonder why not just increase the dose. However, increasing the dose will bring about higher risk of toxicity, more challenging vector production, and increase the manufacturing cost. And recently, 2 patients died in a high dose group of the AAV gene therapy clinical trial. So that's why we developed a new system. It's called a PS gene editing. And what is this system? So it's a CRISPR-based system. We use Cas9 to create double-stranded break at the intron 1 locus of albumin gene. Then through HDR, the splicing acceptor cDNA protease sequence will be integrated. Also NHEJ, the whole donor vector, including homology arms and ITR, will be integrated. However, due to the interaction between splicing donor and splicing acceptor, the fusion transcript will be the same. So exon 1 of cDNA and the cDNA. And this part encodes a signal peptide and will be cleaved thereafter. So the mature protein will be our therapeutic proteins only. And based on this mechanism, we have this study design. We have 2 constructs, cDNA plus guide RNA and Cas9. We package them into 2 separate AAV8 vectors, IV injection into mice. And then therapeutic cDNA will be permanently inserted into the albumin locus of hepatocytes only. And hepatocytes will secrete therapeutic proteins for cross-correction. Then we can look at our data. Oh, before that, I want to talk about our portfolio. So we have in vivo pharmacology, biodistribution, and toxicology studies in animal models. First is wild-type mice and also MPS 1 mice, which has been published in Molecular Therapy earlier this year. And send-off mice, GM1 gangliosidosis, non-human primates, and we have some in vitro pharmacology and safety studies as well. And we can talk about MPS 1 first. So the test article we named the PS822. MPS 1 results from deficiency of alpha L-iduronidase, IDUA, and subsequent accumulation of glycosaminoglycan, or GAG. Typical symptoms include developmental delay, joint problems, and death by age 10. And here's our data. So we have 3 groups, all treated as neonates, high, middle, and low dose. The difference between those is tenfold. And we have untreated MPS 1 and normal mice. And we have MPS 1 treated as an adult as well. And here's the plasma enzyme activity from month 1 all the way to month 10. And here's the log scale. So untreated MPS 1 is green, but you cannot see it here because it's 0. Normal mice between 5 or 6. Low dose 60, middle dose 1,000, high dose 3,000. So basically we achieved more than 600-fold wild type levels in plasma. And we did a comparison. So we have achieved 50 to 250-fold higher transgene expression than the ZFN system. As I mentioned earlier, the ZFN system failed in the clinical trial because of low transgene expression level. But what if we can increase it by 250-fold? We also checked the enzyme activity and GAG storage level in tissues. And first one, we want to talk about IDUA enzyme activity in the liver. As you can, it's log scale again. Normal mice below 10, low dose, middle dose 100, high dose 300 something. So it's 22-fold of wild type levels in the liver. In the brain, and normal mice still below 10, not detectable in low dose, but in the middle dose, it's significantly above background. And in high dose, that's 1.3-fold of wild type levels. What about GAG storage? So again, liver, above 70 in untreated MPS 1, normal mice down here, and we have significant reduction in all three dose groups. And in the brain, untreated 25, normal down here, and we have a dose-dependent reduction. So we achieved a GAG reduction in the brain by 74% and normalized in high dose and middle dose group. We also check survival and behavior tests. So first is a Kaplan-Meier curve from day 0 to day 350. And black line, that's normal mice. Green line, that's untreated MPS1. A lot of them died starting from day 250. And all three dose groups had significant improvements. And we also performed a fear conditioning that evaluates memory and learning ability. So it's a percent of time they're going to freeze upon stimulus. And untreated MPS1, just 25, normalized up here, and all three treatment groups, significant improvement. We also have done POE test, which measures coordination and motor function. And untreated MPS1, this one, and normalized down here, and our treated MPS1. And also Rotarod, 3-day test, normal mice above here, latency to 4. And untreated MPS1 down here, and our treated MPS1, significant improvement. So there's improvements in survival and neural behaviors. What about histology? So we performed H&E staining in the liver first. As you can see in the center panel, that's untreated MPS1, a lot of cellular vacuolation in hepatocytes and Kupffer cells, which is absent in normal and treated MPS1. Also in the brain, in the center, in the Purkinje neurons of the cerebellum, we have seen a lot of vacuolation, which indicates lysosomal accumulation, which is absent in normal and treated MPS1. So this PS gene editing reduced the cellular vacuolation in the liver and the brain. Finally, we have some unpublished data through collaboration with Dr. Walter Lowe's lab. So we use the resting state functional MRI to check the brain connectivity in different regions. So here's a hippocampus, CA1, and normal control, a lot of connectivity between different areas. And untreated MPS1 reduced the connectivity. And in male treated and female treated, significant improvement, restoration of connectivity. Similar observation in ACA. So we have talked about efficacy. What about safety? We have several major observations. First, no vector-associated adverse events, no signs of toxicity, clinical signs, growth or microscopic findings in 1-year follow-up, no increased tumor risk through pathological analysis. And we have also assessed the immune response. So we use the ELISA to measure anti-IgG, IgG against the IDUA, Cas9, and AAV8 capsids. So let me walk you through. We have so many groups. Normal, that's here's normal mice, untreated. And all three red groups, that's a wild type, treated as neonates, and months 1, 5, and 9. Blue, that's adult mice, adult MPS1 mice treated at months 1, 5, 9. So let's focus on the neonates first. So in IDUA, there's no significant difference between these three groups and this group. And similarly, no significant difference between these three groups and this group. Also here. So there's immune tolerance of Cas9, IDUA, and AAV8 in neonatal mice, which is understandable. And in adult mice again, so in this one, no significant increase, but in terms of AAV8, there's a significant increase in terms of antibody. So there's immune tolerance of IDUA and Cas9, but not AAV8 in adult mice, which is interesting. And another thing is that we notice there is an outlier. That's the same mouse, and also the same mouse. So this mouse has antibody against IDUA, Cas9, and AAV8. And we are analyzing data from that mouse right now. We also checked AST and ALT level, which are indicators of cellular immune response. So again, we have so many groups: wild type untreated, wild type treated as neonates, and MPS1 treated as neonates, MPS1 treated as adults, untreated MPS1, But the conclusion is short, no significant difference between any groups. So there's no increase in AST and ALT level after the PS gene editing. And we checked the albumin level because we hijacked albumin production to generate therapeutic proteins for our case. And we're wondering whether that will affect albumin production. And the short answer is no. So untreated MPS 1, treated MPS 1, untreated wild type, treated wild type. No significant reduction. So summary of MPS 1 study, we have achieved up to 250-fold higher efficiency than the ZFN system. And it leads to significant enzyme activity, GAG storage reduction in the brain, as well as improved neural behaviors, histology, and brain connectivity. So this liver targeting strategy can efficiently treat neurological diseases. And it's well tolerated. No adverse events, no signs of genotoxicity, no increased tumor risk. And it seems like we have immune tolerance of Cas9 and IDUA. And back to this portfolio. So we cover MPS 1 mice, and we have published another study in set of mice in gene therapy, but I don't have time to talk about it today. So GM1 gangliosidosis mice, we have a new mouse model, we have some data, and we're going to publish it soon. And we can talk about our results in human hepatocytes because the mouse genome sequence and the human genome sequence is different at the target locus. If we want to treat human patients, we need to redesign the guide RNA and the homology arms. So we test that in human hepatocytes. We test a lot of guide RNAs, and fortunately, we find one that can mediate efficient cleavage at the target locus. And then we transfect human hepatocytes with Cas9 donor and guide RNA, and we use the primer in the donor construct and one primer in the human genome sequence, and perform PCR to confirm integration at the target locus. We found this band showed that there's an integration, and we also measure enzyme activity in supernatants and cell pellets of edited cells, and there's significant increase. Finally, we have done GUIDE-seq, which is an unbiased off-target analysis in a genome-wide manner. So GUIDE-seq detected 120,000 reads at the target locus, but no off-target reads. Then we can get back to our portfolio. So we have covered our in vivo pharmacology, biodistribution, and toxicology studies in different mouse models, and also our data in human hepatocytes, pharmacology, and safety, basically unbiased off-target analysis. And acknowledgements, my collaborators from University of Minnesota and Washington University in St. Louis. And I'm not sure whether I still have time to take questions.
We've got maybe 30 seconds for questions. So I don't see any questions in the question answer panel. Just a reminder to people, if there are any questions, you can type them into the question and answer panel. Otherwise, each of the speakers are going to go to their breakout session after the next talk. So please visit those and feel free to ask your questions to the speakers there.
Okay.
Okay, thank you, Leo. So our final speaker this morning is Dr. Lalita Baylor, also from the University of Minnesota, and she will be describing a different approach for treating MPS 1 using lentiviral transduction of hematopoietic stem cells.